Hello, my name is Vincenzo Interdonato, and I am going to present about octopus skin and their suckers. Yes, suckers are the official scientific name for them. There is no other name. Those little guys are pictured right here. We'll talk about the structure of those a little bit later. And then we have the chromatophores, which are the specialized pigment cells in octopuses. And yes, it is octopuses. It is a Greek and Latin word, which means it is pluralized as one. So over here we have the chromatophore. It has a pigment granule, and that holds all sorts of pigment. And what ends up happening is there are smooth muscle cells that release um, this pigmentation as the octopus needs. What's pretty cool, it's controlled by the central nervous system, as well as each individual limb of an octopus has nervous system capabilities as well. Here's a 3D model of the sucker of the octopus. We have the acetabellum at the very top, and then we have the funibellum down here. Couple different. We have a half 45 or half of it, and this is what it would look like fully. At the top here is where the it would connect to the tentacle itself. So this is how the sucker uh, conforms in order for the octopus to grab hold of something. So first it flattens and conforms to the shape of the surface and performs a flat seal. The muscle in the cerebellum region contracts and generates a reduced pressure in water. And then the continuation of the sealing of this upper chambers continues to protrude downward, which strengthens this and continues strengthens the grip by continuing to reduce the pressure. And then the muscles in the cerebellum continue contract, which is the bottom region, and causes the equilibrium of the pressure to return, and then the suction will release. This specific suction and action is helped strengthened by the elastic modulus of each region of the octopus sucker. So the cerebellum is that top region, and it has a much higher elastic modulus, almost over double of the infundibulum region, which means that the acetabellum region is much more difficult to mold and shape. The infundibulum has the elastic modulus of about 7.7 kilopascals, which compared to the cerebellum is much easier to model. Um, Here we see that it's actually one of the softest biological materials. It's equivalent to the Saglinte morate mesogilia, which is a jellyfish, which is very, very soft and can conform very, very easily. This elastic modulus is described as Young's modulus in a lot of text, but they just use elastic modulus in the resources I was using. So some applications of the sucker research. So there's a picture of the softball robotics, which is featured here. It uses soft sucker technology and the robotic arms to grab very complex objects that regular robot hands and things like that struggle to grab. We have some medicine applications as well. So from medicine delivery patches where they're using it to make sure that the patch doesn't fall off, tissue grafts, and they're even using some surgical tools that utilize this similar methodology in the octopus sucker in order to make sure that they're doing more precision work. As well as continues in advances in just material science and such as like adhesion materials with enhanced grip and durability that they're not seeing in other objects. Some challenges in this research. It's difficult to study specimens, and they can only perform some of these tests on deceased samples for like reasons such as animal cruelty and things like that. These muscles are controlled by the nervous system, and it's difficult to understand this like control with deceased samples in the octopuses. This They use samples that were caught for like eating and food in order to perform these tests. There's even a further complexity because, like discussed earlier, the arms have autonomous control and coordination of the suckers. So that means that as this animal lives and traverses through the environment, the muscles and things like that that coordinate these suckers can be even more complex. So these relationships are not 100% understood, but based off of the pure mechanism and the elasticities, these are the numbers that they were able to confirm. Lastly, I wanted to mention a little bit about the chromatophores because they're pretty cool. They're use the smooth muscle cells that we were kind of talking about a little bit earlier to open these pigment sacs inside of the cells. These are a couple different ones like together. And then there's a sudden and rapid change in the constriction of ink sacs, which results in some unsteady diffusion of the ink. And then here we have the Flick's second law, which we could technically, we had some concentrations and temperatures and things like that. We could definitely use it with this error function like we discussed in class. And then we can potentially solve for the concentration using these chromatophores and the diffusion of ink, which would be pretty interesting. I couldn't find a lot of research, so this is definitely potentially a place we could research an application of things further. Thank you.